Thank you. Finally, it's my turn. So, good afternoon, everyone. Today, I'll talk about our GEM study. So, our GEM study have two things. The first one focuses on searching for novel genetic and molecular features to improve MS outcomes. And the second one focusing on unraveling the interplay between Epstein-Barr virus, genetic genomics, and host T-cell immune regulation in MS. Now, because these two things share the same acronym, and that's GEMS, so we use this acronym for our study. And our study was designed to address the following clinical problems. The first clinical problem is diagnosis. Now, this figure is showing the average time from first symptoms to diagnosis of MS by different criteria. Now, as you can see, even based on the latest McDonald criteria, the average time to diagnosis MS is still 4 years, so which is still quite a long time. And one of the key criteria used to diagnose MS is that the neurologist needed to see a physical damage in the brain as shown in this brain imaging. But as the damage already happened in the brain, the process may be irreversible. So secondly, people who are diagnosed with MS also face an uncertain future as the clinical course of MS is highly variable and we are not able to predict how will they progress over time. So this figure is showing 3 different types of MS clinicals. So the blue line represents the classical relapsing remitting MS, and the green line represents those who started with relapsing remitting MS, then converted to secondary progressive MS. So the red line represents progressive onset MS, which basically the disability slowly progresses over time. Now, for relapse onset MS, some people develop severe disability within 5 years of disease onset, but up to 25% showed no demonstrable disability after 10 years. The third clinical problem is treatment. Now, there's no cure for MS. Despite these different treatments have been approved to be used in the MS clinics to treat MS, so these treatments are not able to prevent the disability progression in the long run. And these therapies also vary in their efficacy, side effects, and risks. So therapies are applied by trial and error with uncertainty that the right therapy is being used for long-term benefit. I will using the Royal Hobart Hospital MS clinic as example. So every year, the clinics implement therapy changes for about 200 people with MS. So the goal of study is to achieve major breakthrough in the field of MS research, to provide better understanding of the human biology of MS, what triggers or causes MS to start and continue to progress, to develop biomarkers which enable early MS diagnosis, better disease course prediction, and a more efficient treatment strategy to reduce disability or cure. So we'll achieve our goals through the following 2 things. The first one is searching for novel genetic and molecular features to improve MS outcomes. So we probably have the experience that sometimes, our GP would ask us to take a blood test in order to say whether we have particular disease. So for example, diabetes. But so far, we don't have a blood-based test for MS. Why? Why we don't have such biological features indicating MS onset and progression? So you probably heard of the story about the blind and elephant. So same for MS, because MS is a very complicated disease, and most of the time we study this disease by testing a particular hypothesis that look into one angle of the disease. So this work lacks a full picture of MS and its development. We probably know that the DNA is blueprint of our body. But if you look at this cell diagram, you'll notice that the different levels of molecular events happened. For example, the transcription of the genes and the generation of the proteins, among others. I'm using this as an example to demonstrate if we can get a comprehensive different level biological data from our body, it will provide a better understanding of MS onset and its progression. So the approach to analyzing the data set spanning different aspects of cell biology is called multi-omics. And multi-omics covers different levels of cell biology, including genomics, which study the complete sets of DNA, all the way to glycomics, which study the carbohydrates, what we call the glycans, that are attached to the proteins to influence its functions. But to comprehensively measure, because its molecules are so tiny, to comprehensively measure them is very challenging, and it really requires new development in technology and innovative approaches. So over the past few years, we have generating the multi-omics data step by step using a longitudinal study we call OSLON study. So the OSLON study has followed more than 250 participants with over 15 years follow-up. And we have the blood and serum samples collected baseline, 5 year, 10 year, and 15 years. So this number is basically showing the available sample that we have for measurement. And we have generating the genomics, epigenomics, and metabolomics data. And finding from our work are published in some of the top journals, such as the Brain. But because of time today, I'm not able to go into details. But the key conclusion from our work is that by measuring different level biological data, we are able to narrow down the candidate list that can be used as a biomarker for MS. So in the future, we plan to using innovative approaches to generate the remaining biological data, such as proteomics and glycomics. And just before the lunch break this morning, we actually got some good news about the success of one of our applications submitted. So, but because it's still under embargo, I'm not able to provide more details. And we also submitted a major grant to NHMRC with outcome available around later November. So hopefully we have some good news. By generating a comprehensive biological data, we hope that we'll have a better understanding of MS onset and progression. And of course, finding from our work will have significant clinical implications, such as novel insights into disease biology, novel disease biomarkers, and drug repurposing and development. Now, the work I presented mostly focusing measuring the data from our own body. In the reality is that we are living in a world that's surrounded by different viruses. So our immune system are constantly fighting different viruses. An epidemiology study has been shown that infection with a particular virus called Epstein-Barr virus may be the key driver for the development of MS. And therefore, the second part of my project focuses on EBV research. So what is EBV? This is what EBV looks like in the microscope. And this is showing the structure of the EBV. So EBV is a double-stranded DNA virus. And the total length of the viral genome is roughly 170,000 base pair. And then this double-stranded DNA is then wrapped up by different virus proteins. So EBV is one of the most common human viruses and infects 95% of adults worldwide. And most people get EBV in early childhood, and the majority experience no symptoms or only mild symptoms such as fatigue or fever. And in some cases, EBV can cause more serious illness such as gaggle fever. So the primary EBV infection is typically transmitted saliva, and that's why MS is called the kissing disease. And even EBV infects our immune system and remains there for the rest of our life. So what do we learn from pandemic is that probably we now have better understanding that COVID have different strains. And for example, we probably know that infection with particular COVID strain, let's say Delta, will have more severe symptoms comparing with infection with Omicron. So same for EBV, because EBV is also a virus, and EBV can also classified into type 1, type 2. And within each type, multiple strains have been identified. So there are no differences in physical properties between EBV strains with different genetic variations. And genetic variations in the EBV genome are likely to result in differences in disease development, such as MS. To test that, we assembled a team with diverse skills. So this including immunologists from Burgerfer, geneticists from Genome Institute of Singapore, statistical geneticists from the University of Queensland, and we also have researchers from our MS Research Flagship, and we have 4 people as associate investigator, including 2 people with MS, and they are Anna and Julia. I think they are here at this meeting. So last year, we secured a major grant funding from Medical Research Funding to continue our work. And the first aim of the study is to identify EBV strains and the variations associated with MS development. So to test that, we'll recruit 420 people with MS and 420 people without MS. We'll collect saliva samples. The reason to do that is because the virus is shading via saliva. And then we'll extract the DNA. We're using the sequencing and the genotyping technology to get the genetic variations within the EBV pathogen genome as well as host genome. So we'll be able to compare the genetic differences between MS and control to identify EBV strains and variations. The second aim of the study is to evaluate the functional impacts of mass spectrometric EBV strains and variations on T-cell immunity. So to do that, we'll recruit 40 people with MS. These are the people with no disease-modifying treatment, and 40 people without MS. And this time we'll get the blood samples because blood samples have different immune cells, and in particular, we'll focus on the immune cell called T cells. And because T cell will respond differently when infected with MS-specific EBV strains or normal EBV strains. And from that, we'll be able to know what kind of T cell or combination of T cell that contribute to MS development following EBV infections. So the significant impact of project including we will identify if there are EBV genetic variations and or EBV strains that lead to greater MS risks. And we'll generate library EBV references genomes specifically relevant to Australian, which can be used to inform EBV vaccine design. And lastly, our finding will provide invaluable insights into potential refinements of the T-cell therapy that our team has developed. And we work closely and collaborate with our consumer communities, the important part of our project. So I will use an example to demonstrate a significant impact. So these two lovely ladies and Julian, they worked as an associate investigator on our EBV project. So when we plan to recruit a participant, they suggest that we use this title, "Let's Split in the Face of MS." At that time, we're not sure whether we can get approved by the ethics, but it turns out, they approved that. And it's quite a huge success because within 7 days of our advertisement, we have received more than 150 participants to complete a survey to join our study. This figure is basically showing, the participants from different states. And because of such a large number, of involvement, we literally have to put our advertisement on hold just to process this, samples. And earlier this year, we were very lucky to have Meg join as an associate investigator on our idea grant for the multi-omics project that I just described before. So hopefully we have some good news later this year. And so the work I present today are actually as a result of hard work of team's work. So I want to thank you every team members, but I also want to thank you the funding body for supporting our work. That's including MS Australian, and I must say, and Medical Research Funding, Royal Hobart Hospital, and also I also would like to thank all the participants who joined the study. So if you have any questions or you're interested or you want to be part of this exciting journey, we do have a trading table over there, so feel free to talk to us. Thank you. I'm happy to answer any questions.